This academy, conceived by a few very visionary people, is progressing. Probably have heard about this academy for young scientists, which is sort of thought to be a sort of college according to the English system, where students are taught both by the university and by their colleges. And I very much support this enterprise. Okay, I very much support this because I think this is one way to draw students into science and medical science. And what I have seen over the past 2 years, it developed from a vision into something that is soon going to be in brick and mortar, if I may say so. We have many virtual institutes all over the place, but my experience with the virtual institute, nobody knows where they are, who is responsible, can't meet them. So I think the idea of having something in brick and mortar is exactly what is needed, and I wish you all the best, or wish us all the best to that this is going to be a success. Now, listening to the mentors' talks today, probably most of the students, especially the very younger ones, I don't see them anymore. I don't want to. Probably they had a hard time today, but nevertheless, the mentors' presentations were very interesting, and I don't want to overload you with too much data. After that experience, I sat through all these lectures, and it was almost like a scientific conference. So I'll try to stick to the basics. Also, I see that the youngsters, the secondary school pupils are not around anymore. They were pretty lost, I must say, at the end of the first session. So I'm going to talk about a theme that is thought to illustrate that, first of all, in contemporary research, the most important thing is to develop methods, new methods. This gives you an advantage over your competitors. And secondly, that you get unexpected results with new methods which the method wasn't designed for. So I'm going to talk about first single channel recording on a very simple level, and then I'll show you how, by using the methodology that we used for recording single channels, we have a new access to functions of single nerve cells and of nerve cells in their natural environment, which is in the cortex a so-called column. The cortex is organized in small repeating units called columns, and these columns are part of cortical circuits, and I'll cut this short, the cortical circuits. So at the end of the lecture, you should know what an ion channel is, what are the functions of cells that are electrically important, and finally you should know what is the function of particular cell types in a cortical column. I hope I can make things still digestible at this time of the day. So, first, as Gabor said, we were awarded a Nobel Prize. This is Erwin Neher and myself for this type of recording. Now, what is this? This is a recording of current through a patch of membrane when the membrane is excited. And what you see here are this is one of the very first recordings. These are step-like transitions between two and three levels. the interesting thing in this slide is this one. This signifies or represents 10 picoampere, and I'll show you what a picoampere is. But it meant at the time that we were more sensitive in recording currents by a factor of 100 to a factor of 1,000. Now, in real life, if you do an experiment, you'll see traces like this. So this is was supposed to be an animation of these currents that you record from a biological membrane when in a patch of membrane ion channels are activated. So this would be the baseline level, and this would correspond to opening and the closing of a single ion channel in this membrane. Now, we were awarded the Nobel Prize for, I was awarded the Nobel Prize together with Erwin Neher, and recording single channels is all very nice, but you want to learn something about. So it was the first proof of the ion channel concept of ion transport. Ions can be transported across a membrane by basically two mechanisms. One is opening a pore, and ions will flow down their electrochemical gradient. The other mechanism is what's called a transport mechanism, where a protein picks up one or two or three ions, changes its conformation, and transports the ion to the other side of the membrane. Now, if we look at the size of these ion channel currents, we find they are in the order of picoamps. And if you calculate then how many ions are transported, let's say per millisecond, it's 2 or 3 orders of magnitude of anything that has been described in biochemistry as the catalytic rate. I mean, a transport system would be basically a catalytic system, and this system can handle at most about 10 to the, about 1,000 per millisecond. That's about the maximum. And these signals which we discovered were 2 or 3 orders of magnitude larger. Second, it was the first observation in real time of a single molecule in biology. Nowadays, there are many discoveries on single molecules, but these were the first discovery, the first recordings of the transition of a single molecule called an ion channel. And finally, and I'm not going to go into this, by using molecular biology methods, we could identify in the protein that forms the channel particular domains that regulate the ion flow. So using point mutational analysis by picking individual amino acids, changing their side chains, we could modulate the size of the current up and down. So this is at least what the quotation says. Now, how do we do this? This is a schematic diagram. We started to work on the muscle, and you put on a patch pipette. It's a glass pipette that is hooked up to an amplifier in current clamp mode. So the amplifier is organized such that it keeps the voltage. This amplifier by a feedback loop keeps the voltage difference 0. So whenever there is a change in voltage caused by a current flowing through the tip of the pipette, it will recompensate. And this is what we record from a single channel currents. The decisive discovery or the decisive thinking behind this is to reduce the background noise of the recording. And you can see that the background noise is proportional to the square root to the inverse of the resistance. So you have to increase what we call the seal resistance or the leak resistance between the inside of the pipette and the outside solution. And this is done by pressing the electrode and applying some negative suction, increasing R and decreasing the background noise. Now, how do we interpret this? So far you have seen only currents. Now this is a view of the tip of the pipette. In blue you would have the membrane. These are the heads of the lipids. And in this membrane are ion channels incorporated. In this case it would be a representation of an acetylcholine receptor channel. By pressing the tip of the pipette onto the membrane and applying negative pressure, we can suck a small patch of membrane into the pipette. And then we are recording traces like this. You have seen this before. This would be the baseline, and this corresponds to a closed channel. Then all of a sudden the channel opens, stays open, and those flips back into the closed state. The important thing is this one is in the order of 4 picoamperes, and the duration is the order of milliseconds. So our nervous system, and not only the nervous system, but also the muscle glands, operate on signals that are due to the superposition of such single ion channel events. In order to signal a message from your brain, let's say to your toe, it's got to go very fast, milliseconds, otherwise you can't walk. And this is done by action potentials, postsynaptic potentials, and all these potentials are generated by the superposition of thousands of such elementary events. So you can think of a single channel current as the basic building block of the electrical signals in our body. Now I want to show this a bit in more detail. So what is happening when we do this recording? At rest, the membrane current is 0. This would be the 0. This would be in this particular case, an acetylcholine receptor channel protein, and the channel would be closed. It has a narrow portion, which we identified in the protein, and this narrow portion is closed. No ions can flow. In this particular case, the channel is opened by the binding of a transmitter molecule. In this case, this is acetylcholine. It binds to a binding site, and once it binds to binding site, the channel flips into the open state. Okay, so this is illustrated here. What we observe is an increasing current corresponding to the opening of an ion channel, allowing the passage of ions through the membrane. And this is what we're recording here. Now, when the transmitter molecule, acetylcholine, dissociates again from the binding site, the channel flips into the closed state, and this is what we record as an upstroke. That is, the channel is shut, the membrane current flips back to the control value. Now, I promised you to tell you what is a picoamp. A picoamp is a thousandth of a thousandth of a thousandth of an ampere. 1, 2, 3, 4. Okay, so an ampere is the current that you use to, produce light in this room, and it is a millionth of a millionth is the size of the current through a single channel, and by these small signals in the picoampere range, all our electrical recording in our body is generated by superposition. You have larger signals, but they are superimposed by these elementary, due to the superposition of these elementary events. Now, the usual signals you see in the CNS, we're in the nanoampere range, and now we can look up into the femtoampere range, and this was very important because it meant that we can record very small signals characteristic of the central nervous system. The central nervous system was studied mostly by intracellular electrodes. Current measurements were not possible due to the high resistance of the recording electrode. Now, using these patch pipettes, it was all of a sudden possible to look into electrical signaling, not only of muscle membranes, but also to record signals from nerve cells in their natural environment. So I'm going to switch now to a section that kept me busy for almost 10 years, where we tried to find out what are the properties of single nerve cells that might be important for signaling in the intact system. Okay? And what we found is that we can do this by adapting the method that I just described. First, to record from a slice of brain. This is supposed to be a slice brain, and it contains, in this case, schematically, 3 neurons. And what we found is that we can, by applying suction to the internal of the pipette, we get a low resistance access to the interior of these cells, meaning we can record the membrane voltage, and we also can fill the cell with ions or substances which we want. For example, a typical case would be a calcium chelator. Calcium is kept very low in the cell, but we can make it even lower, or we can increase and see how this changes the function of the cell. But I'm not going to talk into that at all. I'm more interested, or I'll focus on signaling between the different compartments of a cell. So a nerve cell is, and you have just heard this in one of the mentors' talks, doesn't have only a cell body. It's a much more interesting cell let's say, than a pancreatic cell. I think at least, because it has these processes called dendrites and axons. On the right part of the slide, you just see a picture of how this is done, how we get access. We clean the surface of the membrane by a slight positive pressure. Then we touch, and then we relieve the pressure and apply either a pulse, an electrical pulse, that destroys the membrane in the patch, and then have low resistance access. Now, what can we do with this? The first thing was to overturn a paradigm, and Gabor alluded to this, namely that the dendrites, the receiving compartments of nerve cells, are electrically inexcitable. These are passive cables, so people thought, which just propagate signals according to, basically, to the cable equation. It's attenuated propagation of electrical signals. Now, Greg Stewart and myself thought, well, let's see. We want to see whether this is right. And what we did is we recorded from 2 spots, or from 2 parts of a neuron, with one electrode in the dendrite and one electrode in the soma. The conventional theory was that only passive spread is, these are passive elements, and there's only passive propagation of signals. This just shows you how we do this during an experiment. We fill the cell with patch pipettes filled with 2 fluorescent dyes, and you can see there's a very nice mixing of these 2 fluorescent dyes, indicating that we are indeed recording from the same cell. So this is all very nice to overturn a paradigm that has existed due to the fact that you couldn't record from dendrites. But what has this any sort of functional meaning? This was the next question I was asking. And we used these dual recordings from 1 or 2 cells to find out what are cellular properties that are due to this electrical excitability. So this is first a description of how we made sure that what the electrical excitability is due to. And what you see here is a dual recording with a somatic pipette and a dendritic pipette. I hope you can see this. Okay, here it's much better, yeah. So we are recording with two pipettes, the way I just showed you, and we are setting up an action potential by current injection. Sure enough, there's an action potential, but in the dendrite, we also see what's called a back-propagating action potentials, or in lab jargon, these are BAPs, meaning that whenever a cell fires an action potential, it travels down the axon. This is well known for decades, but there is a copy of the action potential pattern sent back into the dendrites. So action potentials are generated close to the soma and then back propagate and forward propagate. So whenever there is a signaling, the receiving compartment of the dendrite receives a copy of what has happened. Now, this was intriguing. So we thought, what could be a function? And we found something very interesting, and I'm really proud of that. This is experiments done with Matthew Larkham. And what we found is, and you'll see this in an animation, what we found is that when we combine input to the basal dendrites, to the soma, and to the apical dendrite, in a certain time window, these cells act as a coincidence detector. So who knows what a coincidence detector is? It's very well known in physics. There was even a Nobel Prize for inventing a coincidence detection. Anybody knows what a coincidence detector is? It's very simple. You have two signal sources that converge on an element. In this case, it's a neuron. And when the two signal sources are activated within a time window, the coincidence window, then the output will be much stronger than the sum of the two. Okay? So it is a device to detect coincident events. And this has been used to, for example, measure decay rates in physics. Very easy. You can see when there is a coincidence. And what we discovered is that these neurons, pyramidal neurons, act as coincidence detectors. And I hope my animation works this time, because I want to show you how this works. So we have a recording at the soma and a recording at the dendrite. And we're injecting current to simulate synaptic input by the blue pipette and by the yellow pipette. And if you do this in a situation where the yellow input precedes the blue input, you just get a single action potential, okay? However, when we change now the distance between the time distance between the yellow current injection and the blue pipette current injection, you see what's happening. So, okay, let's do this again. Something is haunting me. Huh? Anyway, what you're supposed to see, let's do one more go. Okay, so what you can see is for a certain time interval, for input to this cell by the blue and the yellow pipette, there is a tripling of action potential. I hope you have seen this, that for a coincidence interval of about 50 milliseconds, when two inputs are coincident, then the output of the cell triples, and sending the message to the target cells that a coincidence has happened. And this is a perfect way to signal to the downstream signals, to the downstream targets, that there is parallel input to the upper part of the dendrite and to the lower part of the dendrite. So whenever there is a burst, as we call it, the target cells of these pyramids will know there has been a coincidence, meaning there is input to two spatially separated inputs, to two spatially separated synapses. Okay, and it's very time sensitive. You have seen they have to occur within about 20 to 40 milliseconds. So it's an exquisitely sensitive device. And just as a sort of a remark, it's been very popular to record electrically instead of electrical signals by signals that rely upon calcium transients. And so far, nobody has been able to reproduce this with light signals. It's just you need the information, or you need the precision of electrical recordings. I had a good talk with Tony this morning. He has the same opinion, that for the nervous system that operates in the millisecond time range, recording of calcium signals is all very nice, but it's in a time range which is not exactly very relevant for acute representations. So the next discovery that we made was that if we combine in coupled cells bursts of action potentials in the presynaptic and the postsynaptic cell, postsynaptic being the target. In this case, it seemed to, in this case, they were reciprocally coupled. And what Henry Markham and myself found is that when we time the action potentials that travel down the axon and that travel up into the dendrite, if the timing is again within a time window of about 50 milliseconds, we can increase the coupling between these two cells. The coupling between cells is usually measured by the size of the EPSP. This would be a control period. Then we would use this coincidence protocol pre and postsynaptically, and what you see is there's a creeping of the EPSP indicating that the coupling of these cells has strongly increased, provided you're timing the action potentials pre and postsynaptic in a proper way. And considering the low frequency that we find in the cortex, I think this is a much more likely mechanism underlying what's called plasticity, meaning that you change the coupling between cells, rather than the usual LTP with 100 Hertz stimulation. I think most cortical cells will experience 100 Hertz stimulation only when they're in a slice and an experimenter applies electrical shocks. So this is a direct consequence of this burst activity. When the bursts in two cells are timed properly, you can increase, and this is not shown, or decrease the coupling between these cells. So the last slide in this sequence then shows what we discovered in vitro. We discovered the backpropagation of action potentials into the dendrite. We discovered that there are action potentials bursts generated by coincident inputs. There's a target cell specific short-term plasticity. I didn't go into that, although I wanted, because this was discovered with Peter Somogy some 20 years ago, almost 20 years ago. And there is what's called spike time-dependent plasticity. This is this phenomenon which we have seen in the previous slide. This is called spike time-dependent plasticity. Now, The big question at the end of the 90s was we have discovered four phenomena. Now, is this at all relevant for the working brain? So it could be very well only an artifact that we see in the dish. So the last almost 17 years I spent to prove that these mechanisms which we discovered in vitro are really relevant for the operation of a functioning brain. And I'll show this in the next slides. Now, if you want to find out whether these four phenomena, backpropagation, spike time-dependent plasticity, and target cell-specific STP are really introduced, you first have to define your object, where you're going to measure this. And we decided we are going to look into this in a particular cortex, which is called the somatosensory cortex. This is a cortex that operates in humans, when you touch something. Without sensory cortex, you cannot feel what's going on. You have no feedback. And this is more important than the visual system for rodents. This is why I chose it. And in order to find out whether these phenomena that we discovered in vitro, we had to reconstruct a complete column. Now, how do you do this? To reconstruct a complete column, you can't do this in one shot. You first reconstruct all the cell bodies, then you reconstruct all the dendrites, and then you reconstruct all the axons, and possibly make guesses about the synapses that connect these cells. This is a very old picture from Ramani Cajal, showing the basic building blocks of a nerve cell in the cortex. These are the dendrites. This is the axon, cell body, and he could not resolve synapses because there was no electron microscope. And he had an intense discussion with his competitor Golgi, who maintained it is a net, but Cajal maintained there are synapses, and he was right. So there are four elements: dendrites, soma, and axon. And what we did is we also wanted to know the functional properties of these cells. So we record from cells, fill them, and reconstruct them, and then register them into a standard Cartesian system in order to build a column. And this is what's shown in the next two slides. First of all, the cortex is not a homogeneous mass, as you might guess from functional MRI pictures, which is just they're all red or blue or whatever. They have many different cell types, and the excitatory cell types fall into 9 classes. They are shown here. They have very colorful names, usually by the shape of their soma and the shape of their dendrites. The most colorful names are thick tufted dendrites. This is a thick tuft, and this is a thin tuft. Now you do this on many animals, reconstruct them, register them, and you build what is called what we call the dendrite column. So it shows that a section through the cortex has about a size of 2 millimeters. It is subdivided in different layers, named from layer 1 to layer 5. Actually, there's layer 6. And you see in different colors the extensions of these different dendrites. For example, you can see that these cells, layer 5B cells, have dendrites here and up here. And as you know from the previous slide, this would be an ideal cell to detect inputs from layer 3 and layer 5B. Now, this is only half the picture because you want to know where is the information sent to from a column. And this is, again, years of work to reconstruct not only the dendrites but also the axons. Now, each of these, this would be a single column that you see here. These are the neighboring columns, and you can see that the axon column is far in excess of what is a dendrite column. Actually, it corresponds to one central column and one surround, and surround columns of 8 accessory or surround columns. So the basic unit of the cortex is not a column consisting of dendrites, it is a column consisting of dendrites plus the axons. Now this is rationalized, and I'm not going to show you this in form of a matrix. One can calculate a connection matrix between different cell types, and recently Oberländer has done it for individual cells. So we have for cell types, we have a 50 by 50 matrix showing the the strength of the different connections based on on these anatomical data. The major conclusion which the students should learn is that a column, as defined by CO staining, is only part of the story. A column consists not of dendrites and somata, but also of axons. If you do this, the basic unit is much larger. It's actually an ensemble of 9 different columns in the classical sense. Now, I want now to focus on one particular cell type, and these are the layer 5B cells. These are the cells that have their somata here, have their dendrites up to here, and these are the cells where we discovered these dendritic excitability. And I'm going to use this now to find out whether there is backpropagation, whether there is spike time dependent plasticity, electrical excitability, and target cell specific routing. Now the first thing we found, well, sorry, I'm skipping ahead. So this is a method. One records in vivo from the somatosensory cortex. The somatosensory cortex is made up of an array of these columns that correspond to, each column corresponds to a particular whisker that is deflected by an piezo actuator, and we are recording from cells blindly, define their functional properties, fill them, that is, we ask the question, how many action potentials do they produce, what is the PSP size, and then they are registered into such a column, the way I showed this before. And I would like to focus on these layer 5 pyramids, because they seem to be the most interesting ones, because they sort of sample the activity in the cortex from all different layers. It's the only cell that samples electrical activity from inputs from all different layers. This is why we concentrate on this. And the major question was, what is the function of action potential bursts? The first thing, which was not very well received by the reviewers as well, was that rather that all these cells in the different layers are basically activated by the thalamus. You've just learned what the thalamus is, almost simultaneously. The conventional picture was the major input is into layer 4, and then layer 4 redistributes the activity into the other layers. This is, at least for this cortex, certainly not the case. For the initial 20-30 milliseconds, all cells are activated more or less simultaneously. Amongst it, also these layer 5 cells. Now, when we record from layer 5 cells, we find two, oh gee, can't see the red. You have to see. Okay, you have to focus on this one. So there are two features that are particular for the layer 5 cells. First of all, the response has a fair percentage, about 25, of what we call action potential bursts. We have learned that action potential burst is generated by coincident input into the layer 5 B cells. And secondly, and this is not shown here, these are the cells that respond most strongly. If you scan through a column and ask the question, which is the cell type that generates most action potentials, it is the layer 5 cells. An example of these layer 5 cells from which we are recording, that's the recording from this cell, and in red you have the occurrence of action potential bursts, and it's much higher than in all the other layers. For example, compared to layer 2, 3, I just read a very beautiful paper by Gabor, but what we find, I think he's under conditions which are really relevant. These are freely moving animals, and I couldn't really make it. But I think from looking at the pictures, it seems that the action potential frequency in these layer 2, 3 cells is definitely lower than what we see in layer 5 B cells. It would be nice if you could record from layer 5 B cells the next time. Okay, it took Gabor 6 years. So maybe in another 6 years we'll have this data on layer 5B cell. Okay, good. So they are characterized by the occurrence of these bursts, which you don't see in the other cells. So what could this be? It could be a way to represent the occurrence of a touch. If all cells respond with the same, or with action potentials coming in a almost random sequence, the target cells wouldn't really know which whisker has touched. So we were looking for cells that have a different response characteristics, and we found that this is indeed the case for layer 5B cells. Now, the data that I showed you from combining anatomical and functional data would suggest that layer 5B cells are activated by the thalamic input plus one or two inputs to the apical dendrite. And this generates action potential bursts, and this is not seen in other cells. So they are particular in signaling to the downstream targets that there has been an input to the apical dendrite via the occurrence of action potential bursts. Now, how is this? How can we relate this to behavior? In collaboration with Christian de Kock, we're using the following paradigm. We ask what is happening during decision making when a rat decides to jump across a gap. Before it does this, or before it turns back, it sort of senses the distance between here and here via the whiskers. If you do this if you film this, you see that there's a continuous whisking, and depending on the distance, it either will jump or it will return. So the question then is, what are the cell types that are activated by this repeated whisking in this particular task? And how is it signaled? I mean, how is it coded? How is the occurrence of a touch coded? And I'll give you a brief answer. So there are, if you look at this behavioral task, there are two phases. One is whisking, that is, the animal just tries to explore the space by whisking. And when it touches, it has an active touch, and the whisker is deflected. You can see this. When it touches, the whisker is slightly deflected. And what we did is we are recording now from different cell types in a column that is related to this particular whisker, asking the question, which cell type is likely to signal the occurrence of a touch. I don't know whether this is just how, I don't know whether you can see this. So this is during an experiment. You can see this is a sort of exploratory whisking, and there are several touches which you can't hear because I haven't hooked up the audio. Anyway, so using this, we were asking which are the cell types that respond to touch. And the answer is shown in the next two slides. We were recording from two types of cells, the layer 5 slender tufted cells, and they are active during whisking, that is during the exploratory phase. These cells are very active, layer 5, and these more or less are quiet. Things change, however, if there is a touch. And this is, I hope, this time the video works, otherwise I'm going to complain. Yeah. Okay, so what you see here, or should hear, is a recording of the voltage, of the action potentials occurring during these blue dots, the position of the whisker, and the active touches. And what you see is that action potentials are generated mostly during active touches, but there are also some spontaneous action potentials. But if you do the statistics, ask where are the touches, where are the action potentials occurring? You relate the number of and the sequence of action potentials to a particular phase, namely to the phase when the whisker is touching an object. And if you do this, you find that this is a recording from a layer 5B neuron, that it is the layer 5B neurons that signals unequivocally the occurrence of a touch. And how does it do that? By action potential bursts. This is from a sort of cartoon from a article that Christian de Kock and myself are finishing. It's just one example, but we have done all the statistics. And what's happening is the cell that responds to a whisker touch are the layer 5B cells, and they respond with action potential bursts. So there's a long way from discovering coincidence detection in layer 5B cells, finding that electrical excitability is also seen in vivo, and then using this particular feature, namely the generation of action potential bursts, to find out which type of cell represents the occurrence of a touch. And for the students, it looks as if it is almost exclusively the layer 5B cells that signal to the rest of the brain that a touch has occurred in this particular task, where the touch is signaled by layer 5B cells, and as a consequence, the animal jumps across the gap. We don't know at the moment what are the features of these responses that finally make the animal decide to jump. I think it's not a single cell, it's an ensemble of cells. So my guess is it's the number of cells that respond with action potentials that finally will decide whether an animal jumps across this gap. But I think we have now unequivocal evidence that it's layer 5B cells that code the occurrence of a touch. And this is most likely based on the electrical excitability of these layer 5B cells and their capacity to detect coincidences. This will be published by a former student of mine, Matthew Larkham. There will be a science paper hope before the end of the year, that shows this directly, that when an animal makes a decision, it responds with action potential bursts, and this is due to coincident input to the basal dendrites and to the apical dendrites, as he can show with imaging. So this is what I have to say. I hope I haven't overloaded the students. Try to keep it very simple. There are a few messages. The main message is, if you want to understand the nervous system like the cortex, there is no way not to go from bottom up. The approach from bottom down is leading to all sorts of theories. Might be quite elegant, this and that and that. But if you want to understand that, you have to understand the elements. This is slightly provocative. Many people will not be very happy, but this is at least my experience. Start with the basics. And this is why basic science has to be represented in your school. Okay? So thank you very much.
First of all, really thank you very much for these absolutely fantastic presentations. And as we mentioned today a couple of times, that the whole idea came from a number, and which number was the 75th anniversary of Albert Sakmann's Nobel Prize in 2012. Then we decided to establish this program, which can help many talented students to achieve their aims. This year was also a great year and a milestone for the foundation, because the government made a decision to support all the initiative. However, as I said, this always comes a number. So our last major year was when Albert Sakmann got the Nobel Prize 75 years ago. This time, again, an anniversary, because Bert Sakmann, for his wonderful discovery, was just 25 years ago. So today we heard the back propaganda. I think it was after your presentation, when you showed about the electrode and touches, I think for us you are the electrode and we are touched and we are stimulated.
Okay, good. Thank you.
Thank you very much for accepting to be the educational director of our schools, and please let us celebrate with you and give it this medal for you to commemorate your 25th year of fantastic Nobel Prize. Thank you very much.
Thank you very much. Okay, thank you. So, thanks. Thank you. Okay.